Today's the topic is counter current mechanism. So the first thing, counter current mechanism means what? So it is a mechanism in which the substances are changed between the loop of Henle and Vasarecta. Okay, so to understand this, okay, now why these things or why the substances are exchanged between the loop of Henle and Vasarecta, or before entering in the counter current mechanism, you should first understand why it is so important. Okay, so this mechanism, you can see it is a part of excretory system. Okay, and it is a very important mechanism which is found only in mammals like us. Okay, now why it is important? See, the significance of counter current mechanism is that it will help you to form a concentrated urine. Now concentrated urine means what? In which you will have less, you can say in which you will have more solute concentration, less water. Okay, so for example, whenever we are in dehydration condition, okay, whenever a particular person is in dehydration condition, it means already he has less water inside the body. So in this condition, he will not allow, his body will not allow more water loss. So he will prevent the water loss. How can he prevent the water loss? So by doing this mechanism. Okay. Now, now we will be first this counter current mechanism significance. We know that it will be helping us to make concentrated urine. Okay, so how can we make these things? And you know, there are certain nephrons. Basically, all of us know that you know the functional unit, structural and functional unit of kidney is nephron, which is actually responsible to make urine. Now, in our body, in our kidney, there are two types of nephrons. Okay, one is juxtamedullary and one is cortical nephron. So first, we have to go with the basic things, that is, what is the difference between juxtamedullary and cortical. So if you can see here, this thing, this one is your cortical and this one is your juxtamedullary. So this is your cortical nephron and this is your juxtamedullary nephron. If you see properly, so here, okay, I hope all of us know that you know what is the basic structure of nephron. So you can see this is the cup-shaped structure which is known as Bowman's capsule. This is two convoluted structure, you can see initially here and finally here. So this one is known as, okay fine, this is known as Bowman's capsule. This initially convoluted structure, folded structure is known as PCT. Then you will see a big U-shaped structure which is known as what? Loop of Henle. Then we have the later convoluted structure after the loop of Henle. You will see DCT. You will see DCT, and then finally you will see there is a collecting duct. So this is the basic structure of nephron. We all of us know. Now coming back to the point, cortical and juxtamedullary. What is the major difference? There is a very simple difference. If you see the loop of Henle, the loop of Henle of cortical nephron is short. In contrast, if you see the loop of Henle of juxtamedullary nephron, that is long. Okay, so it means the short loop of Henle is seen in cortical nephron and juxtamedullary nephron are having long, or you can say the loop of Henle is extended here, big, or you can say a large loop of Henle you can see. Now apart from that, there is one more important thing, that if you see these blood vessels, this blood vessel which you can see now, so here this blood vessel which is coiled around the loop of Henle, you can say, or around the nephron, is known as peritubular capillary. And actually this particular peritubular capillary or the blood vessel which is coiled only around the loop of Henle is known as vasa recta. Remember, vasa recta is the blood vessel or the blood capillary which is coiled around the loop of Henle. Okay, and that is densely coiled around the loop of Henle. Vasa recta, student, we can see only in juxtamedullary nephron. It is absent in cortical nephrons. Okay, and as I told you initially only, that counter current mechanism is a mechanism in which substances will be exchanged between the loop of Henle and vasa recta. So if any nephron is not having vasa recta, that will be not involved, that will be not active during the counter current mechanism. Okay, so only the juxtamedullary nephron will participate in this mechanism, and that is why they are important for us. One more thing you can see here, you can see properly why it is so important. See, majorly this counter current mechanism is happening in the medulla region. So we all know that, you know, irrespective, this is the kidney, the outer part of the kidney is known as cortex and inside part is known as medulla. Now the nephron which is actually present, some part of the nephron which is present in the medulla region, okay, that particular nephron can only participate in counter current mechanism. And as I already told you, you can see here also, or you can see here, see this is your, this nephron which you can see is your juxtamedullary, and you can easily see if this inside part is medulla, outer part is cortex, only the juxtamedullary nephron the loop of Henle of juxtamedullary nephron is actually extending the medulla region. Okay, so the loop of Henle extending inside the medulla region only can be seen in juxtamedullary, and this one is your cortical. You can see a very small amount of loop of Henle is entering the medulla, or you can say none. So major nephrons in the kidney are 85% it is cortical, but still it will not be involving in the countercurrent mechanism. The only reason is that loop of Henle is short and vasa recta is absent in the cortical. It is only present in the juxtamedullary. That is why juxtamedullary nephron will be participating in the countercurrent mechanism. Okay. Now what is the next thing for us? Okay. So I hope all of you understood this much part. Now see. As we all know, this particular thing is now here, which you can see in the white thing. I am drawing this is the renal tubule, and whatever is in the red thing, you can see this is the vasa recta for us. Okay, this is the blood vessel which is going around the, you can see, loop of Henle. Now we all know that initially when the blood will come, and this is the glomerulus for us. From here the blood will be filtered, and that filtrate will be moving in this renal tubule like this. Like this it will go from the DCT, finally from the collecting duct, and from here it will exit the kidney. You can see here also a kind of diagram you can see. So once your collecting duct will be giving the filtrate, that filtrate or the urine will be going down to this calyx. This is the renal pelvis, and from here it will be going from ureter we will have here. So in that ureter it will be going down to the urinary bladder and then outside the body. Okay. So basically we know that the filtrate will be moving in this particular renal tubule like this, and then the blood will be moving in this peritubular capillary or vasa recta like this. Okay, how the filtrate will move? So see, filtrate is moving in this direction. For example, first we have to understand the limbs. Okay, now if this is the loop of Henle for us, if this is the loop of Henle, white thing, so here the filtrate is going down. So that is why this limb is known as descending limb of loop of Henle. Now here the filtrate will go up. So this is known as the ascending limb of loop of Henle. Okay, so this is the descending limb and this is the ascending limb of loop of Henle. Now blood will be going in this direction. It means blood here it is going down. So this is the descending limb of vasa recta, and here the blood is going up. So it is known as ascending limb of vasa recta. This is very important for us. Okay. The flow of filtrate is in opposite directions, you can say, in the loop of Henle. Even the blood is also moving in the opposite direction. That is why the counter current is developed. That I will tell you. Okay. So now first we need to see what is counter current mechanism. So student, counter current means what? Counter current means opposite flow. Okay. It means whatever the substances are moving is in opposite direction. You can see here the filtrate is coming like this, and then again it is going up. It means it is going in opposite direction. It means filtrate is also moving in opposite direction in loop of Henle. That is why counter current will be developed in loop of Henle. And then if you see the blood is also moving in opposite direction. Here it is coming down, here it is going up. It means counter current will be developed in vasa recta as well. Okay. So here we need to see the counter current. Basically the counter current is present where? So first we can say one counter current will be present in both the limbs of loop of Henle. You see the filtrate is moving down here, here it is moving up. It means one counter current you can have. One counter current you can have between the limbs of loop of Henle. Second counter current where you can have, can you tell me? See this thing, if you see blood one, blood is also coming down here and here it is going up. It means one counter current you will see in the limbs of vasa recta also. So you will see in the limbs of vasa recta. Now one more important thing you can see. See here, one counter current can be present here also. See, filtrate is moving down, but here the blood is going up. It means in this portion you can have one more counter current. Okay. So here you can say between the limbs of loop of Henle and vasa recta. So in this case, here you can see that here the filtrate is moving down, blood is moving down. In this region also we have a counter current. And here the filtrate is moving up, blood is moving down. Here also we have a counter current. So it means counter current is majorly present here. Okay. And as I already told you, it is the opposite flow. Whenever the things will be moving in opposite flow, it will develop a counter current. And this counter current is very important. Counter current is very important. Then only this complete mechanism can happen. Now, before going deep, we have to understand one more important thing. As I told you, this all stuff, whatever is the loop of Henle and vas recta, now here downwards we have medulla region. This is medulla region of the kidney and this upper one is cortex. Actually, in medulla region there is a osmolarity gradient. Gradient is there. You can say here we have osmolarity. For example, if I say here, if you write the medulla here. So here I will have maybe 400 osmolarity. Maybe here I will have 600 osmolarity. Here I will have 800. Here I will have 1000. And the highest I will have is 1200. And what is in cortex? Cortex will have the basic osmolarity, that is 300 milliosmol per liter. Okay, 300 milliosmol, that is less I will say milliosmol per liter. So in cortex you have the normal osmolarity, but when you go in medulla region you will see that the osmolarity gradient is increasing gradually. Okay, so it means there is an increasing, you can say increasing gradient in medulla region. Okay, now this actually gradient is also created by certain things that I will tell you, but you just have to keep in this mind that in medulla region the vas recta and the loop of Henle is present here, and here in medulla region we have osmolarity gradient, which is very important. Then only this counter current mechanism can happen. In cortex we have the basic osmolarity which we have in the blood, that is 300 milliosmol. In any, you can say, body fluid we have, this is the normal osmolarity, that is 300. Now what is osmolarity, that also I will tell you. Okay, but now, so I hope this much basic things now we know. Now we can directly understand what is the concept of osmolarity. So osmolarity means what? What is actually osmolarity? Student, osmolarity means the concentration of solute in a solution. Okay, osmolarity means the concentration of solute in a solution. It means if you add more solute, the osmolarity will increase. Okay, so it means it is directly proportional to the solute concentration. Okay, but it is inversely proportional to what? The solvent. This is a very important point. Okay, for example, you can take example also. For example, I have this beaker I have here. Okay, and in this beaker maybe I have some solution in which there is a x amount of solute. So it means now what? Maybe the osmolarity of the solution will be x. If I add more solute, so osmolarity will increase. Okay, and if I add more solvent, so what will happen? Osmolarity will decrease. As I already told you, osmolarity is a basic thing. If you add solute, then what will happen? Then osmolarity will increase. Okay, if you want to increase the osmolarity, two things you can do. Either you can add the solute, either you can remove the solvent. This is also the thing. If you are removing the water or the solvent, then also the osmolarity will increase. Okay, and if you want to decrease the osmolarity, so what we can do? Either you can remove the solute or you can add more solvent. Okay, it means you are diluting the concentration. Okay, so I hope all of you now understood the osmolarity concept. Okay, till now we have seen that what is the counter current and the basic thing of osmolarity, what is the osmolarity, and then we all know that this medulla region is having osmolarity gradient, which is very important. Then only the substances can move in opposite direction. This is a very important thing. Osmolarity gradient is present in medulla region. Okay, and it is very important for the counter current mechanism. Now we will directly go with the mechanism, what actually happens. Okay, so here we all know that the blood will come. This is the afferent arteriole we have. This is the efferent arteriole we have. So blood will be coming here in the glomerulus, and from here the blood will be filtered. Student, urine actually is formed by what? From blood only. It is actually a kind of filtrate of blood. Okay. Now, when this particular thing, when the blood will be filtered down, and this filtrate will be moving downward like this, from the Bowman's capsule it will go to the PCT, from PCT to the loop of Henle, from loop of Henle to the DCT, and from the DCT to the collecting duct. After collecting duct, we all know it will be going in calyx, then renal pelvis, then from here it will go to the ureter, and then urinary bladder, and then finally outside. Okay. Now, here when the filtrate will go down, now very important thing is that till here we have the osmolarity, that is what? 300. Okay, but if the filtrate has to go down, see student, anything, any material or any substance which has to come down, it has to maintain the osmolarity in that region. It means if the filtrate at this point it will have the osmolarity that is 300, but if it has to come down, so it means what? It has to increase the osmolarity. Why? Because here we have 400. Okay, it means when the filtrate has to come down, it will have to increase the osmolarity gradually. Okay, continuously in increasing manner it has to increase the osmolarity. So we'll just see here how what things will happen, and we all know basically that this descending limb is permeable for water. The descending limb of loop of Henle is permeable for water, and the ascending limb is permeable for NaCl. Okay, so here what you will do, when the filtrate will move down and now it has to increase the osmolarity, so he can do one thing, he will remove water. It means to come down, this particular filtrate will remove the water. If water will be removed, it means what? Osmolarity will be increased. Now finally when the filtrate will come at this point, here when the filtrate will come, it means it will be highly concentrated and it will have the osmolarity that is 1200. Okay, here at this point the filtrate will have the maximum osmolarity. Maximum water is already retained. Now when it has to go up, so in this case that is ascending limb, here he has to decrease the osmolarity, because now you see, if you move up, it means you have to decrease the osmolarity. So in this case, he can remove what? NaCl. He will remove NaCl. Now, when he will remove the NaCl, when he will continuously remove the NaCl, then osmolarity of the filtrate in this condition will get decreased. Okay, and that is what is important. See, as I already told you, if the filtrate is moving down, he has to increase the osmolarity. So to increase the osmolarity, he is just removing the water. Yes, that is okay, fine. And at this point, it has to enter. Now, when it has to move upwards, it means he has to what? He has to decrease the osmolarity. So to decrease the osmolarity, he will remove what? NaCl, that is the solute. So now at this point, he will again have a decreased osmolarity. And then we have a DCT. Okay. Very important point, student, that in DCT the osmolarity is 200 milliosmol per liter, which is the minimum. You can say this is the least osmolarity we have in the water. So here we will, the filtrate will move from the DCT and then it will be going from the collecting point. No problem. So I hope all of you know now that how the filtrate is moving in this particular mechanism. See, here it is moving down, so he has to increase the osmolarity. To increase the osmolarity, he is removing the water. Here in this condition, it has to decrease the osmolarity. So to decrease the osmolarity, it is removing the NaCl. And then finally it will go to the DCT and then it can come from the collecting duct. Now what about the blood? What about the blood? Okay, fine, we know that filtrate is moving like in this direction. So fine, it is going down. What about the blood? Blood also has to come down now. Okay, very important thing, student. Why this blood vessel is here and the loop of Henle is here and both are participating? There is a very important point, student. If you want to carry out this counter current mechanism, the very important thing is that the loop of Henle and the vasa recta should be in close proximity. See, as I already told you, the things will be exchanged between the vasa recta and the loop of Henle. So, if you want to do the exchange, these two should be in close proximity. Okay, that is a very important point. Now, when the blood will be moving down, it has to increase the osmolarity. Okay, so if it has to increase the osmolarity, it will take up the NaCl. It will take the NaCl which is coming, which is released by the loop of Henle, and if it is taking the NaCl, it means it is increasing the blood osmolarity. Okay. Apart from taking the NaCl, it is also removing some amount of water. It is removing some amount of water. This is very important point. Okay. See, blood is coming down. Okay, so blood is coming down in this case. So it is taking the NaCl. By taking the NaCl, the blood osmolarity will increase. Apart from that, it will also release some amount of water. See, in both the cases, the osmolarity is increased. So similarly, the blood can move down. At this point, again the blood will have 1200 osmolarity. Now, when the blood has to move up, when the blood has to go up, in this condition, what it should do? So in this condition, it will remove the NaCl. It will remove the NaCl. Student, if it is removing the NaCl, it means what? If you are removing the NaCl, it means you are removing the solute. It means indirectly the osmolarity in this case will decrease. Clear? So when the blood will go up, it will release the NaCl. Clear? And it will decrease the osmolarity, and so it can go up. Now the water which is actually released here can also be exchanged by the ascending loop of vasa. See, if it is taking water and removing NaCl, in both the cases it is decreasing the osmolarity. So this is actually what happens, and there is a very important sentence in NCERT also, that the NaCl which is released by the loop of Henle, the NaCl which is released by the ascending loop of Henle is exchanged by the descending vasa recta, descending limb of vasa recta, and that NaCl is again returned in the medullary matrix by the ascending limb of vasa recta. It means the NaCl which we are taking from the loop of Henle here, the same NaCl I am again releasing in the medulla region by the help of ascending limb of vasa recta. Okay, so this is what happens in the counter current mechanism, and this is very important. Then only this NaCl and the water which you are exchanging between the loop of Henle and vasa recta is very important, because this is the only way how you can transport the things. Okay, in this case you can see student in the filtrate which is going here. In that case you are just removing the water, and this water can again taken by the body, and you can maintain the blood volume, you can maintain the water level in your body. Clear? Now, okay, fine, this all of us we know. Now the last important thing is now this osmolarity gradient which you can see in the medulla. The osmolarity gradient which you can see in the medulla is actually maintained by two things. What? The first one is NaCl, and the second one is urea. NaCl, you can see it is, we are just putting the NaCl in the medulla region. Yes, so it means NaCl is definitely present in the medulla region. What about the urea? So now see, urea you can absorb from the collecting duct. We can bring the urea also in the medullary matrix, and this urea and NaCl both can contribute to the osmolarity gradient. So NaCl, we all know it is coming from here, and the urea can be absorbed from the collecting duct. Now this urea you can keep in the medullary matrix to maintain the osmolarity gradient, or if you want, then you can again secrete this particular urea in the ascending limb of loop of Henle. So this is again done. So in short, urea and NaCl is important for maintaining the osmolarity gradient, and this all stuff is nothing but your counter current mechanism. Now counter current mechanism, now we all understood now how the things will be going in opposite direction, what is taken, what is released. Okay, so this is what is important. Okay, this particular thing, student, this mechanism is very important, because if anyone is in dehydration condition, his body will not allow the water loss. If you want to prevent the water loss, it means you have to extract the water from the filtrate. So it means you are absorbing the water. This water which I am removing from the loop of Henle can be taken by the body. Okay, and then you can maintain the water level. That is very important for us. Is this clear with everyone now? I hope all of you understood this thing. Thank you.